Hello everyone, and welcome to today's keynote presentation, CRISPR-powered biosensors. This webinar is part of the 4th annual CRISPR virtual event. I am Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the right of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Dincer, Junior Research Group Leader at FIT and IMTEK at the University of Freiburg, Germany. For a complete biography on our speaker, please visit the Biography tab at the left of your screen. You may now begin your presentation.
Thanks for this kind introduction. Welcome everybody from Germany, Freiburg. I hope where you are all good. And today we're going to speak about CRISPR-powered biosensors. So I have a little bit whole program starting with the biosensors. What are the challenges right now with the biosensors? Coming up with how to combine these with CRISPR-Cas systems. So let's start. So first of all, I would like to say that sensors actually are going to change to disposable sensors. And what does it mean? So here we need low-cost, single-use, and easy-to-handle sensing devices for short-term or single-shot measurements. So such devices are required since they could transduce physical, chemical, or biological changes in their environment to an analytical signal. And these devices are already actually in our lives, like for example the pregnancy test where we have a single use only and then we throw the device away. And why they are so powerful, and also due to the pandemic, of course, getting so important, because these class of sensors enable us to get the physiological information by anyone, anywhere, and at any time without worrying about the contamination as well as needing a recalibration. So therefore disposable. But of course, disposable devices could be also not only a whole device for single use, they could be also combined with, for example, like the evaluation test. So this could be combined, a digital reader together with a disposable part. And here the examples which I showed were some optical, or based on the optical detection, and there are of course also others, and other prominent detection type is the electrochemical system. And here we have mainly the glucose monitoring systems which are made to break through and come to in our daily life. And I choose here, of course, a little bit more not that usual example with a glucose strip and a reader. So you can combine these meanwhile also as a sugar watch into your watches. But also electrochemical glucose monitoring has also gone another step forward and also has now a variable system which allows us, for example here the Freestyle Libre from Abbott, which allows a variable detection of glucose concentration continuously for 14 days. And here you see also the differences of the devices as well as how they are used in our daily life. But again, the question, why this disposability is so important? What is the drive behind? So of course the most important factor is the economic efficiency. it was and it will be most probably also staying. So here we need in high production at an extremely low cost, at minimum quantities of materials, not only the substrate materials, but also the biological materials for a single sensor unit. But there are also other factors, like now the like Fridays for Future and getting these actions, so forces us to make environmental sustainability available. So we are thinking now not only producing the material and also use it, but also what happens after that. And here, of course, it's some future trends is definitely recyclables are there, but we are now moving on to the biodegradable systems. As I already mentioned, the main factor, one of the other main factor in the diagnostics is the contamination of samples. So reusability is something which maybe adds something to your sensing system as a value, but in the general way, you don't want to measure different samples of different patients with the same sensor, since you are deciding the life or death situations, and if any contamination could lead fatal problems. And of course, the importance of the recalibration. So we would like to have easy system. Everyone at home could be able to use it without any difficulty. So, and if independent of what they study, what they learned, and how much they know, it should be really simple to understand and also done at home or at any facility that we could think about it. Regarding the disposable sensors, I think something which is nice for such an educational talk is how they build. And here we have a business model behind, so which is called razor blade business model. And this is actually credited to King Camp Gillette, who found the company Gillette. And actually it is, as I said, credited because actually he did not invent this, but he was the most successful person who is using this razor blade business model, and actually, it is credited to him. What he invented was actually double-edged safer razor. But anyway, so the thing was actually the company has got an offer in the First World War from the U.S. Army, where he get this contract and made these razor blades, sell to the soldier or give to the soldiers, and of course over the wartime they get used to it, and this habit they brought home. If they came there, they continued to use these razor blades from King Camp Gillette, which helps the Gillette company to get bigger. So I think this sounds maybe a little bit off topic, but I think this is still important to understand how if you are dealing with sensors, how you're going to promote them, what should be the strategy behind to sell or to produce them. And here, of course, the main idea is marketing single-use consumables along with a non-disposable, low-cost or even free devices to increase the recurring revenue. And this is, of course, not only for the razor blades. You can find it also with the ink for the inkjet printers. And of course, we have this also, as I show you on the glucose test strips, for example, as a razor blade, or the glucometer as a razor, which is reusable and inexpensive reader. So I think this is also important to understand the basics behind the sensor and sensing devices in overall, since it is important later on to adapt these systems together with the CRISPR. I will finish this part with the requirements of current disposable sensors. As I said, we are going to the inexpensive, sustainable, and even biodegradable materials. Overall, we need high modular devices which have fewer components. So it's important so that you have a really compact device at the end. And of course, again, it should be reliable and low cost, offering and mass production. You need shorter sample result time. So for a point of care testing application for on-site measurements, you need to be sure less than 2 hours. But of course, if it's faster, then it's better. If you have an enzymatic sensor, then it's important you have the fast response time, so that any change which happens in the body you can detect now and not 1 hour later. So the response times are important, otherwise you can miss some events. Overall, it should be easy to use or offer an automatic handling of the samples with minimal user intervention. And at the end, of course, it could be operated with an instrument with a low cost or portable or without. This depends on the application in the final. And of course, an important point, you are moving from the clinical laboratory to on-site, but what you, of course, take with you is the precise results with the international standards. So you cannot be worse while you are just can do it on-site. So you should be at least comparable with the current standards used at the laboratories with your system. And of course, this is maybe something which is, I think, also an important point. Anyway, what kind of sensor you are developing, it's important to think about it like non-technological challenges. For example, if people will accept this in their daily life. So if you are designing a wearable device, would people like to carry these? Or you can make for example, a very good device which can measure very powerful measurements. But the problem at the end will be no one will pay the money that you are offering it, although maybe your device is too powerful for this application. So I think these all aspects we need to be considered before we start building sensors. So what my group in Freiburg are actually working on is we have four different current research areas, which I would like to really shortly go on and then start with our CRISPR work. So first of all, we are working with multiplexing, which means nowadays we do not want to measure a single analyte. We would like to measure different analytes to have a more accurate picture of a disease, or to detect different diseases or different physiological conditions at the same time. And here we are working with antibiotics, inflammation markers, and of course nucleic acids. A little bit side application of what we are working on also bringing these on-site devices a little bit more near to us. So coming to the wearables, we are developing a lab on a bird system where we would like to implement an immunoassay-based hormone detection on freely moving birds. Here, of course, our first model are chickens, and we would like to establish a device which is capable to draw blood continuously every 2 hours and bring it to the sensor, do a classical ELISA on the chip, and the result will be taken via wireless to our smart devices. So this is one of the projects that we are working on. Another one, a little bit away from these, is using optogenetic switches to create dynamic assays. So we would like to use their light-regulated and in reversible interaction by binding them these protein of interest and use the light to bind and release them to create an wash-free ELISA, for example, using a smartphone, which I think this optoassay we will hopefully publish soon so that you can get more information. And the last part is the paper-based breath analysis, which also in my group also done. So this is typical paper-based sensors, which you all might know, but our idea was to use it for a wearable device for non-continuous and continuous and non-invasive monitoring. And our idea was to use the humidity that we are exhaling to make this paper wet for example, in a face mask, and do continuous electrochemical measurements. And here we are working on hydrogen peroxide and glucose. Due to limited time today, I won't speak about it. But if you have, there are also publications in my group, so which you can find. Today, I'm going to talk about what is highlighted, I think the last year, highlighted in Nature Biotechnology last year in the COVID-19 first wave of innovative diagnostics about our multiplex CRISPR biosensor and what we are doing with this. Actually, I was thinking about skipping this slide, but I thought it's an educational meeting today, so I decided to keep it. And let's start why we need nucleic acid testing. So, of course, we would like to prevent, diagnose, and treatment of diseases. So, treatment monitoring of diseases. And why this is important, of course, in case of emergence or re-emergence outbreak of infectious diseases, like the coronavirus that we are, the COVID-19 pandemic that we are living on. And here, of course, as you can see, it's a huge global problem, and we need fast and reliable diagnostics on site so that the infected person can go into the quarantine, and the spreading of the disease could be slowed or even completely disappear. Until the vaccines or whatever the medicine is help us to fight with this pandemic. This is of course the one case, but why my group is started is actually was the novel nucleic acid based biomarkers like the microRNAs, and microRNAs are kind of thought about as a garbage of cells, and currently there is found some correlation of, and not only one microRNA but different microRNA levels to indicating specific diseases. And so this is also why we are interested in at the first side with the nucleic acid testing. And here again, maybe in the microRNAs they are really short RNAs, so and maybe speaking to that, here we have a gold standard, of course, the quantitative polymerase chain reaction, you all might know. So this is the qPCR, and here if you are working with RNAs or RNA-based viruses, you need to first reverse transcript the RNA into the complementary DNA in order to make later the isothermal amplification. But here, it is not easy if you are dealing with microRNAs because, as I said, they are really short sequences. So this is the first problem that you will most probably face. And of course, with the qPCR, you have a time-intensive and expensive method in comparison to the biosensor-based approach. And here, another important thing is limited multiplexing and on-site capability of the qPCR systems is also a problem. And here, of course, we need diagnostic devices for simultaneous, easily scalable, which means you can adapt for new targets easily your system without changing that much parameters. And I think amplification-free, target amplification-free on-site detection of multiple nucleic acids in human body fluids. And that was actually our starting point, and this is also something which we are still, of course, working. And here we started with the CRISPR in, I think it was end of 2017-18. We started with working CRISPR-powered diagnostics, and here in the CRISPR-powered diagnostics, you can find currently two different modes. The first mode, of course, after the isolation and the purification of the sample, the first mode that you could use, and this is also used by Tiana Aran and colleagues at Keck Institute to create the first CRISPR chip, the electrical CRISPR chip, which is based on a binding detection. So they use a deactivated Cas9, which can bind to its target, and in that case they look in the genome some specific mutations, and if it binds to the chip, due to the charges of the sequence, you can see an electrical change which you can detect amplification free in that case. So and this is the first case. So here the cleavage part of the CRISPR enzymes are actually deactivated. The next type that you could use is the cleavage base. And in the cleavage base, actually the Cas is working completely in completely functional. So here, if you start with the target sequence below, so you would see if there is a target sequence, this Cas enzyme gets activated and cleaves all surrounding RNAs except the target RNA. So in that case, the Feng Sang and colleagues used and brought a reporter molecule like the reporter RNA, which can be cleaved and resulting in a signal, and use this visualization technique to see the concentration of the target. And these two techniques can be used for the CRISPR-powered diagnostic, like cleavage or binding. And since cleavage base is a time-depending, of course, it's a time-depending process, so you can achieve higher sensitivities using only the enzymatic activity. So therefore, we decided to go with the cleavage-based systems. Maybe before starting with our work, why maybe CRISPR is getting so prominent, or it's already in the biosensing world, it's also very prominent. So the thing is, we have some different Cas effectors that we are currently using, like Cas13, Cas12, or Cas-C. Even we have a Cas from a bacteriophage found, I think, last year by the Daubner group. And so there are lots of different Cas enzymes, and there are lots of out there. So and the toolbox of CRISPR is really huge, and so it offers many possibilities. And we use this cleavage-based system to create the first electrochemical CRISPR-powered biosensor. And how our system is working, so we have on one side the Cas13a as an enzyme, which you could program by using a CRISPR RNA. And here in the CRISPR RNA, you see the blue part, which is complementary to your target here. So if you are targeting another microRNA, in our case, for example, or other RNA, the only thing that you need to change is this blue part of the CRISPR RNA. And we use also, similar to the Sherlock, a similar or the same reporter RNA with a biosine and fluorescein tag, which we later combine with an electrochemical, with an enzyme to create electrochemical signals. So it's very easy. The only thing that you need to do is mix these three components together with a sample in an epi and wait according to your protocol. It could be 5 minutes to 21 a day at a given temperature. It could be also, it differs by the enzyme that you are using. For Cas13a, it was the 37 degrees. And if you have, in our case, for example, no target microRNA, the CRISPR-Cas does not get activated and the reporter is not cleaved, so that if you are putting in our electrochemical sensor these with a typical, for example, using streptavidin biotin binding, which is blocked with a BSA, and then bind it with an enzyme with glucose oxidase and an antifluorescein antibody, so we can convert glucose to hydrogen peroxide, which we can electrochemically sense later. What happens if we have a target in the sample? So the CRISPR-Cas gets activated and the reporter is cleaved. So I'm imagining always, I gave always the same example. So it's like a Pac-Man going through the epi and clipping all the reporter RNAs, depending on the time you gave it. And here, and of course, the biotinylated part still can bind to the surface, but the signal generating part is just washed away. and so your signal is decreasing if there's a target. So from the assay behavior, you can compare it with a competitive assay, so you get a competitive assay behavior. So this is the whole system, what you need. And as I said, why CRISPR is attracting so many people right now in the biosensing field is exactly the thing that I said at the beginning of this slide. So if you want to target another RNA, for example, the only thing in your whole system is to change the CRISPR RNA. And you can program your device so your biosensing part completely stays as it is. So you don't need to change anything. So you have a very high scalable system. Another thing which is important, and we also use it, is due to this time-dependent cleavage, you can skip, of course, losing some sensitivity, the target amplification, which we also done. Just talking a little bit about the biosensor itself, we have a polymer-based biosensor containing 3 electrodes for the electrochemical sensing, and these electrochemical sensing part and the immobilization area are separated. So we are not doing the complete assay onto our electrodes, but separating it with this, which is called here SP, stopping barrier, so that here our assay is done by giving a droplet to the inlet, which is filled until to the SP part stopping barrier, which is Teflon coated channel. So and then we suck it from the same inlet the solution back, so which allows us to work with different kinds of assay types, and also even whole blood or saliva or any sample type that you can use with using our system. About the assay I won't talk, so it's the same which I already mentioned in the previous slide. But the electrochemical cell later on, of course, what we introduce is glucose, and then give glucose oxidase time to convert glucose to hydrogen peroxide, which we push to the electrochemical cell, to the working electrode, and get a signal peak, which we then later on use for the measurement. Okay, this is the system behind. So what are the results? What can electrochemical CRISPR diagnostic deliver? So we show that it's highly sensitive. So our target was microRNA 19B, which is belongs to the microRNA 17 to 92 family, cluster family. So this is different microRNAs showing a very highly aggressive brain tumor on children. Medulloblastoma is also called. So, and microRNA 19B is a member of this group, and it's highly expressed if you have this tumor. And here we show that our system is calibrated. Our system gives a statistical. This is really important. So we are using statistical with n equal 8, delivered 10 picomole by 3 hours cleavage. So what we can achieve picomolar range without target amplification. And if you compare these with optical devices using CUF13A, we are 5 times better than these devices in the same cleavage time. So as I talk about it, so of course, not only important is the sensitivity, but also specificity, because it's important to differentiate between these different microRNAs so that they not trigger any cleavage. And here we tested our system with microRNA 19B was our target. We tested with microRNA 19A, which differs only a single base, and microRNA 197, which is almost a completely different microRNA. And what we see in the no target, we have, as I said, competitive behavior, so we have a very high signals. And if we have these microRNA 197, we have also very high signals, which means there is no cleavage. In 19B, we see a cleavage as expected. And if we mix these 19B and 197 together, we see again the system works as expected. We also see to microRNA 19A, which differs, as I said, only one single base, we see a cleavage, but still we can differentiate these. And actually, if you know the problem, microRNA or RNA that you are competing with, so you can also build some mismatches in your CRISPR RNA to increase your specificity. In our case, that was not a big problem because microRNA 19A belongs also the same microRNA cluster for medulloblastoma, which means actually this is also highly expressed in our sample. So as I said, one of the main advantages is we have a easily reprogrammable system. So, and to show that, we use microRNA 20A, which we only change, if you can remember, the blue part of the CRISPR RNA, and we directly achieve a calibration curve where we get a limit of detection around 50 picomole for this microRNA, and so you can change changing this blue part allows you be scalable. And yeah. Next thing which we would like to check was actually the cleavage time effect to the limits to the sensitivity as well as the dynamic range that we can measure. So this is the range here between the maximum and minimum, where we can see something differentiate a signal. And here we checked 1 hour to 24 hours. What we see, as expected, the limit of detection gets better. So we get for 1 hour 2 picomolar, to 1 day 2 picomolar, but the dynamic range gets also narrow. And in the one hour we get 18 picomole, but the dynamic range is much more wider. So depending on your application, you need to check the cleavage time and adapt it. Either you want to have a very highly sensitive system, or you just want to have a yes or no answer. So depending on that, you can adapt your biosensing device for this application. In general, our chip-based detection is around 65 cents, and it's also fast compared to the qPCR measurements that we done for measuring the microRNAs. Of course, at the end, getting picomolar sensitivity is not the best sensitivity that you would like to achieve with the microRNAs, but it's the upper limit. So, but we would like to test our system using clinical samples, and we do a little study by using 4 different patients together with a control group and compare our results, biosensor results, with the qPCR measurement. Here, the patient 2 and 4 are in the progressive state of the tumor, so where we expect very high levels of microRNA 19B. And patient 1 and 3 has a surgery, and they are recovering from these tumor, and they are getting, so we are expecting lower levels. And actually, overall, what you can see, the results are very well matching with the qPCR results. We have also we see also in the biosensing part better not accuracy, but better standard deviation. So it looks in general fitting. Only on the patient 1 we see a slight difference for the qPCR results, which are a little bit higher than our patient 1 measurements, which we believe comes from the problematic that the qPCR probes and primers that we use could not even differentiate between 19A and 19B. And as I said, these in our system has at least some specificity against it. So we expect these signals coming from those. Another idea which I was thinking is, so mixing everything in an epi and then putting on a chip. So why we are not doing that on a chip directly? So why we need this? And come up with the idea on-chip cleavage. Is this possible? And we show that it's possible. So the thing which we changed is we build the complete assay into our chip and give the CRISPR-Cas and complex together with the CRISPR RNA and the sample to the channel and let it cleave. So what we see, the system worked. So we see that the system worked, but we had a higher standard deviations and the limit of detection sensitivity is decreased. So this is also something which we are still working on, because this would be a very powerful tool. So you will have one pre-functionalized chip, because we show that our sensors are also stable up to 9 months in a fridge condition after this assay is built there. So, and in that way, you can just take it out, put your sample and wait the cleavage time, whatever, and then do your measurement. So that would be very, very powerful. So one chip for all microRNAs, for example. So this is something, also research, which is still ongoing. And of course, as I said, one microRNA does not help you, and you cannot diagnose anything with that. So it could be upregulated for one reason. So we need to have the whole picture. So we need to at least measure, let's say, 4, 6, or 8 microRNAs simultaneously in the same chip from the same sample. And this is actually also another topic which we are working on, as I said at the beginning as well. So multiplex analysis. And this year we were also able to publish our results. Maybe this is too technical, but I'm trying to explain it. So I showed you our single chip, single channel sensor, where we have an immobilization area, which is shown as black here, and the electrochemical cell. So let's take the incubation area 2 together with the electrochemical cell later on. These two are separated, and our idea was to put a spacer area to another incubation area, which is in that case area 1, so that to measure 2 analytes with the same electrochemical setup by a spacer which separates the signals in between. And if you repeat these, for example, 4 times, then you can achieve 8 analyte chips. And this is, as I said, we already tested in different. This here on the left, you can see the 8 analyte chip, which is each first channel is shown as a blue, and the next one is a red. And we also made, and this is complete a single channel. So we have now different inlets, but in the final, if you are measuring it, it has only an inlet and outlet on the bottom. We also mixed this with a parallel approach, and also made an approach where we have 4 times the same simultaneous after each other. So on the right side, the 4 analyte chip is the repetition of each of the single analyte chip itself. And by using this, we were able, and as I know at that time also, for the first time, we were able to measure 2 different microRNAs using CRISPR, the same Cas effector on the same chip in different concentrations. Here we tested the microRNA 19B in a very high concentration to see very low signals, and no targets to see higher levels of signal in order to check technically if everything is fine. And here on the image you can see the peaks here. Please note here the first peak is important. The next peaks are actually the peaks which is coming from the previous channel all the time. So therefore you see also some following peaks. And this is also something what we are also currently working. So and here I'm also hoping that we will present, we just published another multiplex version of our chips, and now we would like to combine this with the CRISPR to show you the newest results of that. In general, just to give you a slight comparison, and I think here almost you can see I wrote there CRISPR-Cas biosensors, but this is independent of maybe our sensors. Many CRISPR-Cas or the all you can see here as for all general CRISPR-Cas biosensing. What's the flexibility? So you need only CRISPR RNA. So you don't need any specific probes and primers if you are not using a target amplification, which we not using. And it's obviously in a picomolar range. We are trying to also coming to the femtomolar range right now. But even there, we could see, at least in the clinical samples, we could see microRNAs. We can detect microRNAs. You don't need, if you are working with RNAs, you don't need any reverse transcription. Time to result is around 4 hours, depending on the cleavage time, which we use at this work, about 3 hours in qPCR, and for the same microRNAs, we use 6.5 hours. Hands-on time is something which is still we are dealing, because we are not completely integrated system, so we have the biosensor yet. Multiplexing is up to 10 possible with I think with some kind of all biosensors, but this is also for the on-site applications more than enough. I think if you put a control and also a positive and negative control, that would be all completely fine. In qPCR, I think around 4 is the limit. The costs are low. We use only 600 nanoliters of sample to measure and analyze. And as I said, you need a minimally trained personnel right now, but this has the capacity. If it's going to be commercialized, then it could be also easier. What we are also working right now is, as I said, we are bringing the multiplex, now the next level of the multiplex sensors, together with the amplification-free detection. Now we shortened our protocol to 30 minutes, and we are working right now to measure different sequences of COVID-19, where we have an E gene as a first line, and then an ERP gene for the confirmatory, and we have a negative control for SARS-CoV-1, the ERP gene, and a positive control. We are using microRNA 19B, since we have already validated it. Here on the image, it's shown as a single analyte, but we will definitely bring this also with our multiplex chip, since the new generation is already there. Finally, I would like to summarize my talk, since I'm trying not only to show what we are doing, but also give you a feeling what you could do with the CRISPR-Cas combined with biosensors. I would like to give you also, in my opinion, the future perspective. As I said, the CRISPR diagnostics already actually started to move to the point of care. There are companies like Sherlock Diagnostics, Mammoth Biosciences, and they have already point of care kits that you could use. But this is also something which also the biosensors right now is moving. In my opinion, we need to get rid of the target amplification at any cost. So this is either you can use it such a system like the Kiana Aran's group with the CRISPR chip, that you use binding and a very highly sensitive sensor, or like us in a microfluidic channel using some microfluidic protocols to increase the signal. But we need to go down, since in my opinion, if you combine it with the isothermal amplification, then you need again specific primers probes, then you lose the advantage of the CRISPR. And I believe CRISPR has much more potential. And regarding that, actually, if you are targeting, for example, viruses, what you could do, and also the Doudna group already also shown, together with Professor Melanie Oates. You can target the same genome of a virus in specific different areas to increase your target. So you are using 3 targets for one molecule, which increase, of course, the sensitivity of your devices. This could be done for longer sequences. Unfortunately, not possible for microRNAs, but there are lots of different methods to use these amplification-free detections. And I think this will be the next thing, and it's already started again. Of course, but I believe not only the multiplexing to measure different microRNAs simultaneously, what I believe we need to also we will go into the multi-omics, so not only the measuring and nucleic acid, but combining it with an antibody, for example, or to use CRISPR to detect proteins, and not only, and for example, by combining them with some aptamers, which triggers, and if it's bound, it triggers, or it triggers a CRISPR reaction, or it stops a CRISPR reaction, which allows you to detect proteins. I think this could be also an important way where you can think it. Here I choose an example which I found in a soon recently published way, but there are lots of potential out there. I think this is also something which I believe this could be the next thing, and of course wearables. So wearables is in the biosensing the next thing. So it's already started and it will continue the next 5, 10, maybe 20 years. And here I believe the journey of the CRISPR will definitely continue on the wearables, and it's already started in here. There is also a talk coming from my colleague Peter Engayan from MIT, who combined the face mask with a CRISPR-Cas9 Diagnostics. I think this is also the start of the CRISPR into the wearables, but I believe that many works will be some patches or different. Again, here we have a huge potential, very big challenges, and therefore I think CRISPR help us there to solve at least some of these. So with this, I'm end of my talk. This is my group, Disposable Microsystems here, and of course I would like to thank all of my students and also collaborators who are bringing this research front. I'm here the front man to have the honor to speak to you. And finally, I believe in science. So, and this is always one of my last slides. So I believe in science communication is very important. So we need to communicate, and this is one of the current best way is to use the social media. Here you can find me under this link. I would be happy to be connected and share our inside sciences, and happy to hear your comments. Maybe as a last thing, as an editor of a Biosensors Bioelectronics journal, we had a CRISPR-powered biosensing special issue with Kiana, with James Collins, and also the Borjotti Chakraborty. So we would be. I think the deadline is already gone, but if you are interested, just write me. So maybe you can submit as a normal document. So that's also normal submission. With this, I'm end of my talk, and happy to hear your questions.
Thank you, Can, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. We already have some great questions coming in. Our first question, what is the future of CRISPR DX?
I think as I also mentioned, so I think the most important points are right now to achieving some methodologies, maybe combining two different CRISPR reactions together, or doing some other amplification techniques. It could be either microfluidic, electrochemical, or optical, to achieve to bring the sensitivity of whole CRISPR systems without any isothermal amplification. Increase it. So this is, I think, the main point. And as I said, the multiplexing would be definitely something which is also important, and of course variables. So this would be, as I said, I think this will be the next thing. So if you are looking for a variable stuff, so I would go for combining to CRISPR with variable.
Thank you for that. Another question we have, a participant states, this is a great method, but how could you overcome the sample preparation time/complexity? Would it work with different bodily fluids?
So in this work which I show, actually we work with serum samples of children, and we used and sample preparation. So this is something which is important to get rid of. This is also maybe, actually, thanks for this question. This is also very important, and this is also something very important, I think, and also the biosensing community mainly misses this point, the sample preparation, and even how this sample preparation is done, or the blood is administered. These stuff are playing a role on the results. So I think we need here very good protocols. In our system, we said right now we would like to first focus on the sensing part to get the best sensitivity, since we do not directly want to go to the at-home testing, so we would still allow to use it, the sample preparation in a usual lab, but in a longer run, there are also already some approaches. For example, I saw for saliva samples how you could do some sample preparation without easily than we are using using a kit. But I think this is definitely something which is also an important point.
Perfect, thank you. Another question we have, thank you for a very interesting presentation. For the end application, would the enzymatic reaction take place on chip? If so, do you think the Cas enzyme stability could be a challenge and therefore decrease the sensor's shelf life?
And the enzymatic reaction, I think here the enzymatic reaction is meant the Cas enzymatic, because we have also glucose oxidase to signal generation. I'm trying to answer both to be safe on the safe side. So Cas is given the solutions in an all-in-one application, which you see. I think now you can see it again. So in this slide you can see here the glucose is always there, but the Cas will be washed away after the reaction before the glucose comes in. Therefore, I don't think that there will be any problem with that. Regarding in the channel and the cleavage, what I'm thinking, what is happening is what we are currently in this on-chip cleavage living is we have a very short reporter RNA combined with a huge antibody, combined with a huge enzyme. Glucose oxidase is also not small, so we are talking each, I think, 160 kilodaltons molecules, and most probably we have some accessibility problems of the Cas, then some activity problems. This is but something which we are also working on and studying right now.
Great, thank you. There's lots of great questions coming in. We have a couple more here. It says, firstly, thank you for the presentation. I'd like to ask about your biodegradable study. Did you investigate environment effects, especially microbiota?
So I don't know which biodegradable study is meant here. So at the beginning, I believe the materials that we are using will start to be biodegradable in an overall run due to the environmental issue. In our case, our chip is only recyclable, and we do not actually. This is actually regarding the microbiota. We have some activities in another sense using antibiotics, which we are also working on as a group in the multiplex detection of antibiotics, but not for the CRISPR part.
Thank you. Another question, can you comment on the precautions to be taken during miRNA detection and how this will be applicable for point-of-care sensors?
So in the final version, I am not thinking, for example, that you are sitting at home and would like to pipette something, or just it should be much more easier. So maybe the device should be at the end a little bit complicated, so that either you gave your saliva in and that's all that you need to do, and it's the chip, the microfluidic chip is closed, and you have a whole automatic system. where you don't have any access and you don't need to be careful, or you don't need to think about it. But of course, if you are talking in the laboratory, so you have to be very careful. So you need to work on clean surfaces. So RNA contamination is, I think, not only a huge problem for us, but also many people who are working on that field. What we are using in the CAS solution is always we are adding some RNase inhibitor to be just on the safe side, because of course the CAS is also if it's not very good clean, so CAS solutions could also contain some RNases. So just to be on the safe side, we decided to add always RNase inhibitor.
Perfect. And it looks like we have time for one more question. Says, can you please discuss the sensitivity of amplification-free detection by CRISPR without nucleic acid amplification for future clinical applications?
Thanks for this question. So this is, as I said, we are now in the low level of a picomolar range. In the current work, we are now touching in the higher upper limit of the femtomolar range. We have now also some ideas how to push the sensitivity a little bit further with using some electrochemical techniques. Regarding the applications, we are working currently on Alzheimer. So we are looking for different microRNAs which can be found on patients, because in Alzheimer that's a really big problem. So we don't have any good biomarkers to diagnose it earlier, and it's really horrible. And for example, I like always this notice. So if you are already on over 30, it's not you cannot change your future. So either you will have Alzheimer or not. So you can only delay it. So it's a huge problem. So we need biomarkers which indicate it directly earlier ages so that we can find it. And I think here microRNAs could be something which can help us, and this is something which we are working on. Of course, tumor area with cancer, this is also a huge issue which we are working on. Actually, I did not work until the COVID with the viruses. This is of course also something, but also the similar to viruses, the bacteria would be also some problem. And to be honest, I was expecting actually more bacterial cause problems due to the superbugs or whatever than a virus pandemic. But okay, so that's so I think these are the next stuff. And definitely I agree with the question. So sensitivity an issue, sample preparation and another issue. So we need to have easy systems to solve these problems, since as we also saw at the beginning of this pandemic, so the stuff that you need in an enormous, so you need materials, so you need kits, etc. So it's not affordable at the beginning. If the whole world starts to create or needs, even masks is difficult. So talking about not about the kits. So I was really shocked that a country like Germany was not capable of producing face masks at the beginning of a pandemic with such an economy and such a resources. But this is the case, and I think from this pandemic we learned also some lessons, and I hope that this will never happen again.
Awesome. Thank you again, Can, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Any questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years till September of 2023. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and anybody who may have missed today's live event. Until next time, thank you. Goodbye.
Goodbye.